Welcome to week 5. So this week we are finally jumping into our first set of organisms. We will be starting with Staphylococcus, something very commonly seen in the micro departments. So some general characteristics, Staphylococcus are very much gram-positive cocci. They tend to occur in clusters. We will learn about Streptococcus next week. Those tend to be more chains. So staph tend to be more of the clusters. Sometimes you hear them referred to grapes because grapes, when you pick up grapes, they're clustered. So you might hear that kind of reference, grape-like or something like that. But they're nice, big gram-positive cocci. Image gram stain seen here. They're also very much catalase positive. So this is where we're going to first really start delving into some testing, biochemical testing that we perform to begin identifying what we're playing with. And so anytime that we are looking at an organism, we of course gram stain first. We've already learned that. And in this case, it comes up a gram-positive cocci. Once you know the gram stain and it is a gram-positive cocci, the very next test you always perform is catalase. And so staph are catalase positive, whereas next week when we learn about Streptococcus, those are catalase negative. So the catalase will really differentiate between the two groups. So that's why we always perform catalase after we determine it's a gram-positive cocci. So staph, catalase positive. So these are just some mentions here on the left of different organisms, different staphs that you might hear of. See, there is many more than this. There's a lot of staphs out there. You'll run into a lot more than just this. We kind of whittled it down to some of the more prevalent ones to keep in mind for this class. Staph aureus is by far the most that you'll see out of all of the staphs. That is very common, everyday occurrence in the micro department. Staph epidermidis, another big one, not as much causing disease or infection, but more like a lot of contamination at times with that one, but it can pop up for different infections. And you see some others here. So starting with Staph aureus are at the top, the most prevalent out of the staphs. It is a normal flora, meaning again, normal flora means it's found in our body. You know, it does play a role, purpose. Normal flora is bacteria that lives in your body in different sites. They do help play a role with, you know, not allowing other bacteria to take over. They kind of keep the space to themselves, if you will. If you wipe out someone's normal flora, then you're at risk of something else coming in and thriving there that does cause, you know, problems. But the Staph aureus that we have that occurs as normal flora in us is in the nares, so your nostril area, nasopharynx area, your perineal area, and a little bit of skin. Now, there is a lot of pathogenic Staph aureus that we do see as infections. So the Staph aureus we have in us typically doesn't cause a problem unless you become super immunosuppressed and it flares up. Or, of course, if you pick up a different strain of Staph aureus into like a urine infection or a wound infection, something that way. So when staph is past the mode of transmission, it can be spread to normally sterile sites. So like I said, the urine is normally sterile. We do not have bacteria in our urine normally. And so you could cause a UTI or surgical wound, abrasion, anything that introduces it into an area that it's not normally found would lead to causing an infection. So that's one mode of transmission is spread to normally sterile sites. Also person-to-person fomites. So fomites are like non-living objects, like a doorknob, a countertop, you know, toilet handle, anything like that. So if somebody else touched it that had the infection or sneezed on it or whatever, and then you go along and touch it, you can pick it up that way through the air, respiratory, or through unwashed hands. So that's Staph aureus. Staph epidermidis, as it says in the name, epidermidis, very much skin, heavily normal flora of our skin and mucous membrane. So that is predominant normal flora for us. So spread to normally sterile sites, usually through the result of medical devices, prosthetic devices that are being introduced. Staph epi doesn't cause us a lot of problems. Again, it's just a huge normal flora. If we see it pop up, a lot of times it might be just contamination, like when they were trying to collect the wound culture, maybe they swabbed a little bit of the skin, so Staph epi also got on there. And it's not really contaminating the wound, it was just accidental pickup from the site. You know, but when it does cause disease, it's because it was introduced into a sterile area a lot of times as a result of prosthetic or medical devices. So one common example is cardiac like valve devices, things like that. And so then all of a sudden it gets put into an area where it wasn't, and it was just on that device, and then it can thrive and cause infection there. Staph hemolyticus, Staph lugdunensis, very similar to Staph epidermidis. Just not as seen probably as much, but very much just normal flora of skin, mucous membranes, very similar to mode of transmission as well there. Staph saprophyticus is normal flora of your skin and mucosa of your genitourinary tract. That becomes big to remember because of what it typically causes. Staph saprophyticus mainly causes UTIs, especially in women. So it's heavily found in the genitourinary tract. And so if it gets fully put into the urinary tract, then it can cause a UTI. That's pretty much all we see for Staph saprophyticus. Not really much of anything else. It's heavily UTI based because of how it's found and introduced. And so when sexually active women, if they don't, that's why they always say you should go to the bathroom after you have sex to prevent infection. This is one reason. So upon having sex, if it got introduced into there and they don't kind of flush it back out right away, it can hang in that urinary tract and then cause an infection. And then Micrococcus species is very similar to Staph. That's why they're put into this lecture, into this chapter. So Micrococcus species, just contaminants, basically. Very much normal flora of our skin, mucosa, or pharynx. Rarely does it cause infections. So, but you will see them pop up and you'll hear them reference, oh, that's just Micrococcus. And it's typically just contamination on that culture. All right, so a little bit more on Staph aureus, since that is our most prevalent out of the bunch. It is considered the most virulent as a result. And what does most virulent indicate? Really good at causing infection. It has a bunch of toxins and other enzymes and things like that that really help it cause infection. It helps it evade our immune system and really settle in and do what it needs to do to cause infection. So that's what it's always meant by virulence. And so all organisms have some sort of virulence factors. Some have a bunch, some have really good virulence factors, others don't have that much, and that's why they don't cause infection. Whereas Staph aureus do have some good virulence factors that help it cause the infection. So it has a bunch of toxins listed first there, alpha, beta, gamma, delta. They can help destroy those immune cells that are trying to get rid of it. Leukocidin also helps destroy phagocytes. Remember, your neutrophils especially are huge phagocytes. Their role is to gobble up bacteria and destroy them. Well, in Staph aureus' case, they have this leukocidin to help kind of evade and destroy those phagocytes before they can get gobbled up. They will possess the enzyme coagulase, which is a test that we run in the lab. So that's going to become important. Hyaluronidase, that just helps it survive in tissues, kind of invade and really embed into the tissues in the body. And then they have some other exotoxins listed there. This TSST will become big with one of the infections that it can cause is toxic shock syndrome. That kind of plays a role there. So yeah, lots of great virulence factors to help it cause their infections. So spectrum of disease, pretty much almost anything. Staph aureus can be found in pretty much any infection. So skin infections like different impetigo, boils, folliculitis, wound infections. There's also other deep infections. So like bones, joints, organs, lungs. So pneumonia, if you will, there. Bacteremia. So like if it gets into your bloodstream, it's commonly seen that way. It can cause sepsis then. Scalded skin syndrome is a big one with like babies. Food poisoning, toxic shock syndrome. For those, that is basically has always been heavily linked with tampon usage and for women. So it can cause like an all over shock syndrome. So that's why they always say not to leave your tampon in for too long. It can be, really start to cause infection that way if people tend to leave those in for longer than they should. So that's always been a warning that, is on the inserts in tampon boxes. I grew up learning that. I don't know if it's still taught that way, but yeah, so that's always been linked with that. So, and then the list goes on. This is just a tiny summary of what Staph aureus can cause. I always just say it can cause anything, any type of infection in your body. It can be there. UTIs, commonly found with UTIs. That's not listed on here, but it is. So basically any kind of infection will go along with Staph aureus. All right, switching over to Staph epidermidis, the virulence factor here, the one virulence factor you should remember here and why it can cause the infections it does is that it can make this what we call exopolysaccharide slime or biofilm. So it basically lets them adhere to a surface, and then it kind of creates a covering over them, and then they form this like little community in that little under that slime layer on that surface, and it's really hard to get rid of it once it forms that biofilm, that slime. It's really hard for our immune system to try to penetrate through that slime and like get rid of them. So that's why when we said before, Staph epi, pretty much a contaminant, but when it does cause infection, again, it's with medical devices, prosthetic devices. So you see a lot of that, you know, here. We have vascular catheters, cardiac valves, catheter sites, other prosthetics. And that's because when it does get onto those prosthetic devices or medical devices, it will form that layer of slime, that biofilm, and hang onto that surface, and it will basically thrive then, because again, your immune system can't, you know, get through that slime layer. And so that's where it can really pick up and cause that infection. Otherwise, it's typically a contaminant if it pops up in culture. You know, you have to kind of know the source. That's why in micro, it's always important to know the source of where that culture is coming from. When we get a culture in, we don't just say wound. We'll say like a spot on the body, it's wound, or in this case, like, you know, whatever they are sending us, we want to know the source, because then we can kind of link it to where we know bacteria can thrive and where they like to be. So that's another reason. So this is important, like to know the virulence factor, because it leads right into why it causes the medical device, prosthetic device infection. So I think this, yeah, I have a nice example of a really good electron microscope 3D kind of image view of a biofilm formation. It's really interesting. If you ever go online and like Google biofilms and like look at the images, it's quite crazy where you can find biofilms. You know, Staph epi is not the only bacteria that can form a biofilm. There's lots of organisms that are capable of doing biofilm formation. I know I think it's a school in Montana or Idaho that I found a website of theirs at one point that they had a whole biofilm site, and it was really interesting that, you know, the study and the deep dive that they did into it, because biofilms are also environmental. It's not just medical that we're concerned. You can find them heavily in the environment. It's one reason I don't use sponges in my kitchen. I never use a sponge in my kitchen because they love thriving in sponges and forming in sponges. They like moist, wet environments. And so a kitchen sponge, ideal for biofilm formation, because they're like, this is great. And you can't see it with the naked eye. They're there though. And they're in a lot of other places. You might get kind of turned off if you go too deep, you know, keep it in mind where they can form. But that's one reason I don't tend to use sponges. Or if you do, it's okay to use sponges, just replace them often. Same with those loofahs that people love to use in the bathroom, in the shower, those loofahs that hang by the little rope. They always say replace those probably once a month, because they will form in there. And you think of it's kind of counteractive to what you're trying to do, which is clean your body. But then you have like, if you keep that loofah too long, it's forming biofilms inside of it, because again, it's a nice wet environment for it. So it is really crazy. Kind of, it's fun to see the images, but kind of like, oh, like maybe you don't want to see them always. So, all right, Staph hemolyticus, Staph lugdunensis, very not certain on the virulence factor, but again, very similar to Staph epi. I don't really focus on hemolyticus and lugdunensis as much, but keep it in mind, it's definitely some staphs that you're going to see pop up, very similar cases to Staph epi. And then Staph saprophyticus, not totally great on defining the virulence factors here, but again, found in the genitourinary tract. So the disease that it's been heavily linked to is UTIs, and especially in more sexually active women. But again, you can find it as UTIs in anybody really, but that's who it tends to be found in. Other types of infections, not as common. And then Micrococcus, again, very low virulence, meaning doesn't really cause much infection. It's very, very rare that Micrococcus would ever cause infection because it has such low virulence. So pretty much just contamination. But again, we do see it pop up as contaminants in the micro labs. So that's why it's important to know what it is. All right, so that was a lot of bigger ones. There is one more that I'm actually surprised I don't have on here. I thought I did. There is one other staph called Staph intermedius. So if let me see here. I'm just going to do this in real time. Oh, I copied it as an image. That's why it's not letting me play with it. But if I write it out here, so there is another one called Staph intermedius, and I believe this is probably on your homework assignment. And maybe I have it mentioned further down in the, I think I have it in the lab testing area. That's where I have it. This one has been found as oral flora. So it's normal flora kind of the oropharynx, so more oral, like especially of dogs. And so the infection has been linked to is dog bite wound infections. So if an animal bites you, the normal flora can be passed into that wound. So Staph intermedius would be one of those, and it could cause an infection of that wound area if it's not kept well cleaned and addressed right away. So that's kind of been the big focus here. I think I do have it mentioned again in the lab testing area, but I wanted to mention it here as well, that will be one that we will keep in mind as well, and that I do want us to know, Staphylococcus intermedius. So keep that in mind, and we're going to again see it here, I think, in a little bit. So let me go back to where I was. Okay, lab diagnosis. So very much in the micro department, we're kind of generally using a flowchart process. And again, we talked briefly last week about molecular, or 2 weeks ago about molecular stuff. Molecular is the wave of the future for lab testing. With DNA testing, they can test it much quicker in a few hours versus when we work this up in a traditional manner, it takes 2 days, 3 days to finalize everything. So it takes a while to do anything. Whereas with DNA testing, we can see it within a couple hours. It's much better for patient care. So we're focused on the lab diagnosis in more of a traditional manner here. Again, when you get to, actual clinical departments, you're going to see a lot more molecular based items, molecular analyzers. But again, those analyzers don't always run correctly. They don't always give you everything that you need. Sometimes they give you a result and you're like, that just doesn't make sense. And maybe you want to do the traditional testing to verify things, or maybe your analyzer is down and it's not working. That happens. So you have to go back to the traditional testing on the bench. So that's why we still have to know the traditional way of testing. Your board will test you on the, biochemical results, knowing those results. So you do have to keep all this in mind. Even though we're advancing in more, machines and analyzers. So, all right. So we do use in the traditional method a flow chart process. So we always start with gram stain. In the case of this, once we know it's a gram-positive cocci, again, it usually is in clusters. The next step would be catalase, which I think I have on the next slide. For augurs, we always, of course, also read our colony morphology on agar plates. Staph aureus is a gram-positive cocci. Of course, it's going to grow on blood agar plate and chocolate agar. Both of those are supportive augurs. They will grow almost everything. Blood agar doesn't grow all the fastidious organisms, those pickier ones. Chocolate will, though. Staph is not a picky organism. That's why staph will love to grow on blood agar. It grows on any supportive agar. Of course, it's also going to grow on PEA and CNA. Remember, PEA and CNA are good for your gram-positives, so it will definitely grow on those. There is another agar that we have not learned yet called mannitol salt agar, which is seen here. That is a good agar to specifically look for Staph aureus. So Staph aureus loves to grow in the presence of salt and ferments what we call mannitol, which is a sugar. So because it can ferment that mannitol, it will look like a yellow colony when it grows on that mannitol salt agar. Anything else that tends to grow on mannitol salt agar will not be yellow, does not ferment mannitol. So they will be, regular colored. They'll leave it red. So this image shows you up here in the area of 1, they streaked out Micrococcus. In the area of 2 over here, they streaked out Staphylococcus epidermidis. And then down here in area 3, they streaked out Staphylococcus aureus. So this is just a good representation. This is a mannitol salt plate. They incubated it, looked at it the next day, and they were to see if the colonies turned yellow or not. So in the case of number 3, very much yellow, and that's because it was Staph aureus. In 1 and 2, Micrococcus and Staph epi do not ferment mannitol, so they do not change the color of that agar in those colonies. So again, Staph aureus loves salt and loves to ferment that mannitol sugar. So that's why it changes into that yellow colony. So important to remember there. All right, so as I said, once we know the gram stain, you kind of do your colony morphology, you run the catalase test. Again, the purpose is to separate if you have Staphylococcus or if you have Streptococcus, because both are Gram-Pos cocci. So it just helps you start to figure out what you might be playing with. So catalase is an enzyme that is found in the Staphylococcus species. That enzyme will basically break down hydrogen peroxide into oxygen and water. When it does that, you get bubbles, and bubbles are a positive result. So really in the lab, all you're using as your reagent is hydrogen peroxide. That is the reagent. You essentially place a drop of hydrogen peroxide on a slide, on a microscope slide, just like this. You put a drop there. You pick up a little bit of your colony off the agar plate, you mix it in, and you're looking for bubbles. If it bubbles, as you can see on the right here, it's positive. You would call that catalase positive. If it does not bubble, it's catalase negative. It's as simple as that. I wish every test was this simple. It'd be so nice. And you guys will definitely be doing this in your student labs. It's an easy test to run, and it's an easy test to interpret, thankfully. But again, all Staph species are catalase positive. So you see that bolded there. Just one note, there is initials, the chemical initials for hydrogen peroxide, in case you see them on a test question, is H2O2. H2O2 is hydrogen peroxide. So again, that is the reagent that we are using in our catalase test. So after we know it's catalase positive, the very next test we run is coagulase. And that is because Staph aureus definitely has the enzyme coagulase in it. I mentioned that back up here in your virulence factors on Staph aureus. So remember when we went through the virulence factors, I said there is this enzyme coagulase, and it'll become important later. It has that enzyme as part of its virulence factors. The other Staph do not have coagulase, typically. There's a couple, but most Staph do not. And so because Staph aureus is our big prevalent organism out of the group, we want to see, do we have Staph aureus or do we have another Staph? So that's why we always perform coagulase next, is to differentiate if it's Staph aureus from all the others. So Staph aureus actually makes two different kinds of that coagulase enzyme. It makes what we call a bound coagulase, also known as a clumping factor, which that was listed on the virulence factors, or it makes what we call a free coagulase. So we can actually run two different forms of the coagulase test looking for that enzyme. In the bound one, that means that it is bound to the bacterial cell wall and it reacts with fibrinogen in your body. So fibrinogen, as if you learned in hematology, fibrinogen makes a fibrin clot. So in this case, you can see what it's kind of called clumping, clotting, if you will. So in this test, it's basically looking to see, will there be a clot formation with the test? I'd say, yep, coagulase positive. So it causes a change in that fibrinogen, so the bacteria cells clump. And so that would show you a positive if you get that. In the free, it's looking for that enzyme. And again, that enzyme can help form a clot, but it's more in a free form enzyme setting, not bound to the bacterial cell wall. So let's take a look at kind of the image here. So we have two types of tests. The slide test looks for the bound coagulase. So we see the slide test here. So when you take the reagent, you mix in your colony with it and you swirl it around on the slide test, you will look for agglutination because that clumping. So again, it's clumping factor agglutination. It's saying, yep, that clumped up and it formed, and saying, yep, that bound coagulase is here. So that is positive when you see that white agglutination, the clumping there. Negative, it just doesn't change at all. In the tube test is looking for the free coagulase. So the upper one here is positive. It forms that gelatin-like clot. And in the bottom one, it just no clot at all. It stays liquid. So both can be tested for. We can test for either type of coagulase. Often you might see labs do a confirmatory. They might get a positive on one, but they still want to confirm it with the other, so that you can see where they will carry both types of coagulase and use them kind of as a backup to confirm the other one. Or maybe they get a negative on the one and they're like, this seems weird. This seems fishy. I feel like it should be positive, and they still want to test the other one. So they can run both as like more of a confirmatory process. So again, so this is showing that the slide test, the clumping factor. This is a very grainy image. I apologize for that. But again, it should form the white clumping at the bottom here to be a positive. It can be a presumptive that maybe does need to be confirmed with the tube test. And then they run the tube test as a confirmatory, positive. Not every lab will do this, of course. Some labs will just have one type on hand. Some labs will always do this. Every lab will be a little different in how they like to work up their stuff. But it is a note, and it is definitely test questions to hear sometimes of confirmatory tube coagulase testing. So just keep that in mind. But in the lab, you will definitely be doing, I think you guys will probably be doing the slide one in your lab, in your student lab. So after the coagulase test, I probably should have put this above. This is going back to the colony morphology. I probably should reorganize my slides a bit here. But colony morphology, most staphs tend to look very similar. They tend to be medium to large colonies. They tend to be like what we call creamier, but the tone of them, the color of them might be a tiny bit different. They might be white to gray. Some might have a yellow hue to them. So they it'll depend. They look very similar amongst each other. Now, hemolysis, some will have hemolysis, some won't. So starting up here, Micrococcus, this one is one that can kind of be a lot of different colors. It can even be an orangey, a pinky, tan. So it can have quite a few different colors to it, depending on what Micrococcus is there. But again, typically contamination, but it's still important to know how it looks on an agar plate to separate it from something else. Staph aureus is medium to large, creamy colonies. Typically have a yellow hue to them. Not always, again, not every strain of staph stays exactly looking the same. We always get those ones that don't like to look like the others, but otherwise, they typically might have that yellow hue or at least be a cream color. And then they very much are beta hemolytic a lot of times. Again, there are some strains of Staph aureus that maybe aren't beta hemolytic, but overall, they typically are beta hemolytic. So what I always like to teach students here to help remember the yellowy kind of look is aureus actually means gold, golden. And if you think back to your periodic table of elements, AU in the periodic table is gold, is for the element gold. And the word aureus itself means golden, something to that effect. So that's why it was named that, is because of that yellow hue that it can take on the colony appearance. So remembering it can be a yellower hue, definitely beta hemolytic. I have it bolded. Staph epi can be small, medium, gray, white, non-hemolytic. So it's gamma hemolytic. Sometimes it can be a little stickier, not always. Staph hemolyticus hominis is another one I haven't mentioned yet, but it's there. Again, there's a lot of staphs. I said we can't learn them all. Legionensis is described there. Saprophyticus also can be kind of more buttery-like, typically white, but again, it can have that yellow hue to it at times as well. So, you can see that they range anywhere from creamy to a yellow hue on a lot of them. And then here's what Staph aureus you kind of see. I wanted to show that yellow hue on letter A here, showing, and then again, that classic beta hemolysis. Again, you hold the plate up to the light to read the hemolysis through. So you should see that in lab that it has a nice beta hemolysis there. In the upper right image over here, it's showing Staph aureus yellow colonies right here, compared to here's the Staph epidermidis white gray colonies. So that's kind of a nice difference in the color tones. When we describe these, we're not, everybody's going to see color a tiny bit different. You just do your best description as possible. But you can definitely see these are medium to larger colonies. When we get to next week with the streps, they are very small colonies compared to staph. Staphs tend to be a lot larger colonies than streps. So, but it is nice to see that kind of that yellow hue to the white gray look of the Staph epi there. And then again, you have your gram-pos cocci clusters at the bottom. All right, so differentiating gram-pos, catalase pos, cocci. So again, staph and micrococci are all catalase positive. Streps next week will be catalase negative. We will also learn more about Enterococcus next week. They tend to be a weak positive. We still group them more with the streps, but we will very much learn about Enterococcus next week. They can be a weak catalase positive, but Staph and Micrococcus, very much catalase positive. If you were to perform an oxidase test, which oxidase is more of a gram-negative test, we will get into oxidase and microbiology too. Actually, no, we'll get into it later this quarter because we do have some gram-negatives we learned this quarter. We will learn more about the oxidase at that point. It is usually used for gram-negatives. We don't typically test our gram-positives on it. If you were, you would find Staphs are oxidase negative, but Micrococcus is oxidase positive. So that is one way you can separate out a Micrococcus from a Staph, is by using that oxidase if you wanted to. And then how they like to grow. Staphs can pretty much grow in almost any environment. They're your facultative anaerobes, meaning they can grow with oxygen, they can grow without oxygen, they don't care. They will pretty much thrive in any environment that way. Micrococcus is a strict aerobe. It has to have oxygen to grow. And then again, once we know it's catalase positive, we can go down and do the coagulase test. As I said, Staph aureus, heavily coagulase positive. That is a big feature of it. There are, again, a couple other Staphs that like to be coagulase positive at times. One of them is that Staph intermedius. So you see V here for variable. Some strains of Staph intermedius can be coagulase positive, and some strains of Staph intermedius are coagulase negative. So in that case, we can run another test called the PYR test. And PYR stands for the reagent that's used in it. It's like pyridonyl. It's a long word. I can't even remember the full word right now offhand. I would recognize it if I saw it. The word itself starts with PYR in it, but it's pyridonyl, something, blah, blah. If we ran that, which you can run this in your student lab, Staph aureus is PYR negative, whereas Staph intermedius is PYR positive. So that will help separate between if it's Staph aureus or Staph intermedius. So that is important to remember. And then again, I have it listed here, Staph intermedius, dog bite, wound infections. But again, so it does talk further here. Most labs don't identify the coagulase negative Staph to species unless it's coming from like a prosthetic device, a catheters, or from UTI, because then you're getting into Staph epidermidis, Staph saprophyticus, Staph lugdunensis, hominis, hemolyticus, all those other ones. Typically, you don't always, unless, you know, you definitely need to see if it's coming from a sterile site. So urine, anywhere that you might have put a catheter prosthetic device where you should not be having bacteria, then we do want to identify down to the species there for sure. And so doing that, you have what we call those coagulase negative Staph, meaning all the others besides Staph saprophytic, or besides Staph aureus, which is coagulase positive, all the others are typically under the coagulase negative title. Again, intermedius is variable on that. But the big ones out of the others are Staph sapro versus Staph epidermidis and others. You can run what we call a novobiocin disc. And again, this is something you can do in the student lab. So you can put a disc on an agar plate with the organism that you're trying to figure out, incubate it, and look at it the next day for what we call a zone around that disc. We learned about zones with the antibiotics last week. So same thing. Once you take it out of the incubator, you're going to look for a zone or not. If there is no zone, meaning the bacteria grew right up to that disc, it's considered resistant. That is Staph saprophyticus. If there is a zone, and so there's an area of clearing and it's the right amount, you know, you have to always have a right measurement, you got to measure it, then it's considered susceptible. And so that would be Staph epidermidis and all the others. So Staph epi, Staph lugdunensis, Staph hemolyticus, all the others are susceptible. So novobiocin is a great way to differentiate between Staph sapro from all the others, especially if you're looking at a urine culture. Because again, Staph sapro is a pathogen for UTIs. It doesn't cause much problems for anything else, but it definitely causes an issue with UTIs. So we would want to use a novobiocin disc on those urine cultures if we think we have a Staph saprophyticus. So that is really important to remember there. Okay, some other quick mentions here. I'm sure you guys have all heard of MRSA, MRSA, which stands for methicillin-resistant Staphylococcus aureus. This has become quite prevalent, unfortunately. It is a drug-resistant strain of Staph aureus, and it's very much spread through community contact with infected people or things that they've touched and that haven't been well cleaned. So anytime you have people that live in close proximity with each other, think nursing home environments are prone to this. You have a lot of people in a very close proximity in that environment. There's also things like gymnasiums with the equipment in gymnasiums, especially like think wrestling mats. If they're not getting cleaned properly, there's a risk of getting it through things like that. Locker room environments, stuff like that. It could be an easy spread for community contact. So what happens with Staph aureus that it becomes drug resistant? And so methicillin resistant is saying that that Staph aureus cannot be used for any beta lactams. You cannot treat that Staph aureus with any of the beta lactams. So your penicillins, all of those others that we learned last week in the beta lactam group, it is resistant to it. So there goes a huge group of antibiotics that we used to use. So in this organism that has become resistant, they have a gene called the MEK-A gene that is responsible for being resistant. So that MEK-A gene will encode this penicillin binding protein 2A. That protein has a low affinity to bind with the beta lactam antibiotics, which allows it to just continue replicating like normal and become resistant to those beta lactams. So it basically just says we are not allowing that beta lactam to bind onto us and to break us down. We are low affinity for it. We don't care about it and we can avoid it, kind of thing. So we do test for MRSA in the lab, and commonly now, again, it's done through molecular, which molecular can test genes. And so in that molecular testing, they are looking for this MEK-A gene. And if the MEK-A gene is positive, it says, yes, you have an MRSA strain of Staph aureus. You have a drug-resistant strain of Staph aureus. There is also now vancomycin-resistant Staph aureus called VRSA, and that is because when we found MRSA, because we can't use the entire beta lactams, they often went to using vancomycin to treat the MRSA. So vancomycin, again, is a type of glycopeptide. And so they would use vancomycin to treat that MRSA to get rid of that infection. And then as a result, through time, that those organisms learned how to also become resistant to the vancomycin that was used. It isn't as prevalent to find VRSA, thankfully. They're still pretty rare to be VRSA, but it's becoming more and more. This, again, is why there's so much talk about being very conscious of how you prescribe antibiotics, how often you give antibiotics. Is it truly a necessity? Because, again, they were thrown around so easily, and then we started discovering, oh, these bacteria are becoming resistant because we're just exposing them so much, and they're figuring it out. They're like, oh, we're evolving. They evolved too. So they were figuring out how to avoid those antibiotics that were commonly being used. And so that's, again, why we have a huge, there's committees around this on proper antibiotic usage and dosage and everything. So when they have VRSA, then we have to use a combination of some other antibiotics called linezolid and daptomycin. I don't think I said the linezolid right. I might have. It's hard. But again, not as known to be spread through contact, at least with the VRSA. If somebody does have it, it's not spread through community contact like the MRSA. The MRSA is definitely spread through community contact, through close proximity. That is why anytime somebody in the hospital setting, if they are a known MRSA infection, they go into contact isolation. We very much do not we don't put them with a roommate unless the roommate also has MRSA. You have to glove up, gown up, to protect yourself. That way you're not carrying it from others to other, patients, that kind of thing. So thankfully at this point, VRSA is not known to spread through contact like the MRSA, but it could happen. So it's something to keep an eye on. Now, there are screens that will be done. A lot of hospitals have implemented MRSA screens. So if they have a patient that is going to be admitted into the hospital, they screen them for MRSA because, again, if they did have it, they don't want to just put them in a room with another patient, then that patient also gets it, and then the next, and they're trying to reduce down how much MRSA is getting passed in that hospital. So they will implement screens for their patients that are getting admitted. So the way that they do this is they collect it from the nares, because think about it, your Staph aureus is normal flora of your nares, of your nostril area. And so by, collecting the Staph aureus out of there, they can use what we call a chromogenic agar plate. They will streak out onto that agar plate and incubate it. And the next day they look for a certain color. So chrome means color. So those chromogenic agar plates, they're used in a bunch of different ways. There's one specifically for MRSA, but there's ones for a lot of other ones too, if you're wanting to screen for other things. So in this case, on this chrome agar that I'm showing in the image, they are looking for mauve colored colonies. And if there is mauve colonies, it indicates you have a possible MRSA present here. So you have a Staph aureus with the MECA gene present. And so then they would know to put that patient into contact isolation, keep them away from other patients, and make sure that healthcare workers also protect themselves from passing it along. So that is one way to do screens. Again, you can also screen through molecular testing. It's cheaper to run it through chrome agar. Molecular can be quite more expensive, but it's quicker through molecular versus, on the chrome agar, waiting for it to grow and then looking at it. So, but that is a common thing to do and to screen for, is that MRSA, because it's become so prevalent. All right, so that is your info start into our first group of organisms on Staph. Again, it will help when you get into the student lab and can play with some of these tests. We very much follow different flow chart processes. You'll kind of get the hang of that. Gram stain, if it's gram-positive catalase. If it's catalase positive, it's probably a Staph. Then we want a coagulase test to see if it's Staph aureus or another one. If it's coagulase negative and it's coming out of a urine, maybe it's Staph sapros, so we can do a nova biasyn disc. So it's very much a flow to figure out what you have to help identify these organisms. So you'll be practicing that a lot more in the student lab. All right, that is it. I hope everybody has a great rest of their week.